Hello boys, welcome back to another interesting, very interesting session of video tutorials. And this session is meant for class 12th biology students. And this is meant for 31st August 2020. And in this session, we are going to study a new unit called biotechnology. And this is the life of for all of us. And this has got a very bright future. And these two chapters, chapter 11, biotechnology, principles and processes, and biotechnology and its applications, if you focus on these two chapters, you not only get to say 10 to 12 marks in your CBSE exam, that's different story, but this gives you career in your life. Who knows, some of you may become biotechnologists and you may discover many things that will save our lives and possibly give solutions to many unsolved problems in our lives. Hence, focus on this, and whenever you are studying, keep your mind, keep your heart and study. So what is that we are going to discuss? As I said, biotechnology is the new baby of biology. And I am using deliberately the word baby because it is to yet develop and become an adult. It takes time, but then the baby has got very rich potential. So basically the term biotechnology is derived from the fusion of biology and technology. Biotechnology means any technological application. See, we are using technology and we are applying that technological application that uses biological systems, living organisms or their derivatives to produce, modify products or processes for specific use. So this is how we define biotechnology. And what is that we are giving? Yes, biotechnology is the use of living systems and organisms. So we can use the entire bacteria, entire virus or systems of it to develop or make useful products. What is the purpose? Why are we using this? To make useful products or to develop further. Any technological application that uses biological systems, living organisms or derivatives thereof to make or modify products or process for specific use. This is the definition of biotechnology. And European Federation of Biotechnology, we say EFB, has defined biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Now, the oldest form of biotechnology, we have been using breads, curds with the help of microorganisms. You must have seen your mummy dropping a sample of curds into the milk and keeping it overnight, and next day you get curds. How is that the curd has been formed? Your mummy may not be knowing, but all of you know it. It's because of the microorganisms, lactobacillus. Okay. Similarly, yeast also is present, and in the bread also you have seen in our mess also. Plus in your own home and in many Iyengar bakeries you must have seen the process of formation of bread. Compulsorily fermentation process, yeast will be there. So here the oldest form of biotechnology is being used at home, and then similarly in some of the small industries you must have seen application of fermentation in production of wine and other alcoholic beverages is also a biotechnological technique. So in all this the microbes are used. So what are the principles involved? Two important principles that enable development of modern biotechnology. What are those? One, alteration of chemistry. That is, alteration of chemistry of DNA and RNA. We know in our previous session, molecular biology, you know everything about DNA, its chemical structure, everything. So if we can change, if we can alter that chemistry, for example, we said the nitrogen base pairs, adenine pairs with the thymine. Suppose we remove thymine and put something else. So we have altered the chemistry of DNA. Similarly, RNA, In RNA also you have studied the types of RNA, messenger RNA, all that, codon, anticodon. If you change something there, then it is the changed version is to be introduced into the host organism to change phenotype. Phenotype, remember, physical appearance of an organism due to its genotype, we say, and that is genetic engineering. So we said it includes techniques to alter the nature of genetic material of host organism by introducing foreign or recombinant DNA into the host organisms, thereby changing their phenotype. This is called genetic engineering. Normally engineering students or rather engineers, what do they do? What is their work? Construction, building up buildings, materials, roads, everything. No? Here what is that the biology engineer is doing? He is constructing new genes by changing or altering the chemistry of DNA and RNA. So since new genes are being constructed by this biology student, and we say genetic engineering. And second one is chemical engineering, that is maintenance of sterile ambience means environment, to enable growth of desired microbe or eukaryotic cell in large quantities for manufacture of biotechnological products like vaccines, enzymes, beverages, drugs, etc. Already we are waiting for COVID-19 vaccine. The moment we get the COVID-19 vaccine, I'm sure all of you will be back to the school. And it is not a joke, because the vaccine is to be tested at 3-4 levels, that's going on. And then the vaccine should be tested for any possible side effects, that is also going on. And then the real challenge is supplying to 130 crores of Indians. So it has to be large quantity. We require large quantity of COVID-19 vaccine. So naturally it takes time. So these things come under chemical engineering. So chemical engineering basically involves maintenance of sterile. Sterile means no infection is there. Microbial contamination free conditions, no microbial contamination, that allows growth of only the desired microbes or eukaryotic cells in large quantities for the manufacture of biotechnological products such as antibiotics, vaccines, enzymes, medicines, hormones, etc. So these are the two principles involved in biotechnology. One is genetic engineering, second is chemical engineering. So genetic engineering is the technology involved in the synthesis of artificial genes, repair of genes through fusion, deletion, inversion, shifting of genes, products of recombinant DNA, and manipulating them for improvement in human beings, plants, animals, and microbes. So this is what they do in genetic engineering. An important aspect of genetic engineering is rDNA technology. So you got DNA, you insert, first thing you identify the DNA, extract the DNA, identify particular segment, remove that, in its place you put another portion of DNA, then we say it's a new DNA. So we say recombinant, combination, combinant, recombination. So recombinant DNA. So an important aspect of genetic engineering is recombinant DNA technology. Recombinant DNA technology is employed for combining DNA from two different organisms to produce new recombinant DNA. Okay, this is the purpose of genetic engineering. So the three basic steps in creating GMOs, genetically modified organisms, we say. Okay, what we call GMOs. First one is identification of DNA with desirable genes, desirable what we wanted, desirable genes. Second, introduction of the identified DNA into the host. And third, maintenance of introduced DNA in the host and the transfer of DNA to its progeny. So this is what we do in genetic engineering. So basically, asexual reproduction in organism preserves genetic information, whereas sexual reproduction leads to variation. It includes undesirable genes with desirable genes. So genetic engineering, the purpose just now we discussed is to isolate, introduce only one or set of desirable genes without introducing undesirable genes. That means it is selected genes. Techniques of genetic engineering: creation of recombinant DNA, use of gene cloning, and gene transfer to host. Now, recombinant DNA or rDNA, alien DNA means new DNA, cannot multiply itself until it is integrated into host genome. When inherited into host DNA, ability to replicate due to origin of replication, ORI, host DNA, initiates replication. Alien DNA linked with host DNA replicates itself, multiplies itself along with host DNA. This is called cloning. So, gene transfer to a host requires one vector. Somebody should carry that. Commonly used vectors are plasmids. So, what is a plasmid? Plasmid is a small, circular, double-stranded, self-replicating, extra-chromosomal, we can say DNA, extra-chromosomal DNA. So, that is plasmid. First recombinant DNA was constructed thanks to two great scientists, Stanley Cohen and Herbert Boyer in 1972, by linking gene encoding for antibiotic resistance with the plasmid of Salmonella typhi. So, what a great experiment, and this has given rise to a new branch of biology called biotechnology, and biotechnology is going to rule the world in future. Isolation of desirable genes, that is, what is the gene that we wanted? Antibiotic resistant. And cutting out piece of DNA from a plasmid responsible for antibiotic resistance, that involves molecular scissors. Now see, whenever we want to cut something, we require scissors. But here, what is that we have to cut? DNA, portion of DNA. So naturally they are called molecular scissors. DNA molecule is to be cut. So who will cut this molecular scissors? Who will act as molecular scissors? Restriction enzymes. And desirable gene, alien DNA, linked with plasmid. Plasmid just now we said vector to transfer into host organism. So linking of DNA involves DNA ligase, an enzyme, acts on cut DNA molecules and join their ends. New combination, circular autonomously replicating DNA created in vitro. This is how recombinant DNA is synthesized. So rDNA transferred into E. coli, DNA replicate using host DNA polymerase. Multiple copies of antibiotic resistant gene is prepared in E. coli, and E. coli becomes antibiotic resistant. So basic steps in genetically modifying organisms. First, we have to identify DNA with desirable gene. That is the first one. Second one, introduction of the identified DNA into host. And third one, maintenance of introduced DNA in the host and transfer of the DNA into its progeny. So for all this to happen, it's not so easy as we are discussing. We need certain tools. So we say biological tools for making rDNA technology. That